Okay, so I'll
Okay.
introduce Leonie.
Okay.
Okay, so while we wait for the load shedding people to try and get back on, I'm going to introduce our speaker all the way from New Zealand, Dr. Leonie Venter, who attained her PhD in biochemistry at the Northwest University in South Africa. So she's now working as a research fellow at Auckland University of Technology in New Zealand within the Aquaculture Biotechnology Research Group. So it's a very interesting field. She applies metabolomics research to aquaculture industries with the main aim of understanding how environmental stresses affect native aquaculture species and how this knowledge can effectively be transferred to farming practices. So she's looking at specifically at the green-lipped mussel, the Pernalicious. I'm terrible with names like that. The New Zealand aquaculture industry quite a lot, and there's a lot of export value to it. So the climate extremes such as marine heat waves affect the coastal regions of New Zealand, which houses aquaculture facilities and commercial and recreational fishing grounds. So in effect, these changes influence the mussel conditions and threatens mussel survival. So in the study to be presented, that she's going to talk to us about, they were gradually exposed to high temperature stresses, mimicking a 5-day marine heat wave event, and to better understand the effects of the heat stresses on the metabolism of mussels and overall mussel health and survival. And the findings that she's going to talk about demonstrate adaptations to heat stress, including shifts in energy metabolism, alterations in key metabolic pathways, and it is anticipated that the data reported can be used to support aquaculture industry initiatives and policies to evaluate the risks of marine heat waves and improve mitigation strategies. So that is the nutshell that Leonie, Dr. Venter, is going to present. It over to you, Dr. Venter. If you can share your slides.
Mm-hmm.
There we go.
Yep.
And the floor is yours.
Thank you. So good morning or good afternoon or good evening, depending on in which time zone you find yourself. As Shane mentioned, I'm Leonie. And before we start, imagine you were on a game show. Let's say Who Wants to Be a Millionaire? And you are one question away from winning 1 million rand. And you don't have any lifelines left. And the question relates to mussel metabolism and the effect of thermal stress on them. I sincerely hope you never get such a ridiculous question ever in your life for 1 million rand. But this question has been at the forefront of a lot of the research that I'm a part of. And today I'll be sharing some of the findings which are sprouted from this research. So firstly, just a little bit about me. I am a biochemist working within the Aquaculture Biotechnology Research Group at Auckland University of Technology, where we are striving to serve and advance the New Zealand's growing aquaculture industry. Some of the best bits about my job is that I get to work in diverse environments with exceptional people investigating fascinating native New Zealand aquaculture species, such as the green-lip mussel, which is the star of today's presentation. In New Zealand, mussel aquaculture largely depends on seed and feed from the natural environment. Typically, mussel farming is started where spat are grown on long line systems, which are thinned out and reseeded as growth continues, and harvested when a shell length of around 8 to 10 centimeters are achieved. The New Zealand green-lip mussel farming industry is the largest aquaculture sector in terms of value and production quantity, with frozen half-shell mussels, mussel oil, and whole animals mainly exported. Areas such as the Marlborough Sounds and the Coromandel region support the majority of New Zealand's mussel aquaculture activity. As the growing demand for seafood surpasses production at this stage, mussel aquaculture will play an important part in the New Zealand government's aquaculture strategy of achieving $3 billion in annual sales by 2030. But Mother Nature is not too concerned with the government's plans, and climate extremes are affecting all walks of life, especially coastal regions, which houses aquaculture facilities and commercial and recreational fishing grounds. Climate extremes go hand in hand with abnormal weather events, biodiversity and habitat losses. We also experience increased stresses within the ocean, and all of this ultimately results in economic losses. One climate extreme which has been in the spotlight in the last few years are marine heat waves. So what is a marine heat wave, you might ask? So it can be defined as a period of 5 days or more where the sea surface temperature exceeds the normal temperature within that specific area. So the term marine heat waves is very popular for media reporting purposes, as you can see on screen by some of the reporting staff here in New Zealand. So I may not be a climate scientist per se, but the plots on screen give a good illustration of some of the marine heat waves which has been recorded within the last year. So the areas, highlighting areas of the Marlborough Sounds and the Coromandel. So on the y-axis, we see the sea surface temperature, and then from these plots, we see that marine heat waves are becoming more frequent, posing a major threat to the aquaculture industry, where warm water anomalies are causing mortalities and increased diseases in aquaculture species. So how can we measure what is happening on a cellular level in mussels during a marine heatwave event? Research areas with the potential to look at mussel physiology is metabolomics, which enables the identification and quantification of metabolites in a biological system at a given time point to serve as a direct signature of biochemical activity. So multiple metabolomic studies have been used to investigate how temperature affects the greenlip mussel metabolism. For example, in one study where the mussels were exposed to 30 degrees Celsius at 30 degrees Celsius for 60 minutes, we were able to obtain a blueprint of all of the metabolic pathways which were affected by heat stress, highlighting all of the major energy producing systems. In a different study, mussels were exposed to 24 degrees Celsius for 24 hours. And herein, sulfur metabolism was highlighted as a key pathway affected by the heat stress. And also when mussels are subjected to 6 degrees Celsius stress for 24 hours, we see large changes in different amino acids. So thanks to these studies and various other studies, we have a pretty good idea of what is happening on a metabolic level when mussels are exposed to thermal stress. In a nutshell, they activate anaerobic metabolic pathways such as lactate and opines and the succinate pathway to support energy production. You also see an increase in antioxidant metabolites via the glutathione pathway, and branched-chain amino acids are also implemented to support various metabolic pathways as well as immune functions. We've also seen ammonia detoxification via the ureacycle metabolites. But what we don't know at this stage is how mussel metabolism responds during a marine heat wave. So how long does it take for metabolic changes to come into play? And if anaerobic end products are being produced, can they be eliminated or reduced over time? And is metabolic homeostasis achieved via this marine heat wave during this marine heat wave period? Also, at what point during a marine heat wave are mussels the most stressed? And is there biomarkers which suggest adaptation in these mussels? So this study forms a part of a larger program to better understand mechanisms that drive mussel physiology during thermal events. Research linked to these themes are important to build resilience and safeguard the aquaculture industry. This research also supports efforts to strengthen biosecurity management, while also supplying results to help the industry to adapt to environmental changes. The aim of this specific experiment was to investigate the metabolic changes that occur in greenlip mussels when they're exposed to a 5-day laboratory-based marine heat wave. So getting our hands dirty. This is the wet labs at Cawthron Institute, which is the collaborators of this study. And it's a pretty cool system they have down there. So the mussels were placed in these tanks, with each tank with its own water supply and air supply. And then the tanks are stacked on these racks, and which each rack were monitored at a different temperature. It's a recirculating system, so the water goes through filters and back into it all. So all of the dirty work happens in the wet lab, then the mussels go through this little window and it ends up in the dry lab, and there we do the dissection. We did the dissections. So just to put this into context, there were two groups of mussels, of which the one group were kept at 16 degrees Celsius, and they were referred to as the controls for this experiment. And then the second group of animals were ramped up until the target temperature of 24 degrees Celsius was achieved. Then the mussels were sampled for 5 days. During the sampling, hemolymph was collected. So hemolymph is the same equivalent to blood in humans. So the blood of the mussels were collected and placed in microcentrifuge tubes and snap frozen using liquid nitrogen, and then stored until metabolomic analysis were performed. The metabolomics section started with metabolite extraction. We're using a methanol water extraction method, and then the samples were dried and shipped to the Northwest University, where liquid chromatography tandem mass spectrometry analysis were performed using a dynamic MRM database targeting central carbon metabolites. So next, data processing was done and statistical analysis were performed. So based on two-way ANOVA assessments, the following Venn diagrams were achieved. So the metabolites affected by temperature are reflected in the PCA plot with the two groups, where the blue blocks represent the 16 degrees Celsius, mussels kept at 16 degrees Celsius, and the 24 degrees stressor is in the red blob. Then this PCI represents the metabolites that statistically significantly differed due to the effect of time. And here we see that the animals sampled at time point 5 had a there's separation between time point 5 and the other time points, suggesting clear differences in their metabolite profile. But of particular interest for this study is the metabolites that was affected due to the interaction between time and temperature. So the metabolites that differ due to effect of both time and temperature were plotted on a metabolic map covering these main metabolic pathways. In each instance, a plot like this was constructed, where on the y-axis, it is the normalized metabolite abundance, and on the x-axis is just the sampling time, so day 1 to 5. The red lines represent the mussels kept at 16 degrees Celsius, while the blue lines represent the mussels kept at 24 degrees Celsius. And each of the data points on this plot illustrates the average of 3 mussels. So when we start looking at the results, some of the sugar-based metabolites within the heat-stressed group, so the blue lines, the mussels kept at 24 degrees Celsius, they showed a decrease in metabolite abundance at time point 2, whereafter an increase was seen in the heat-stressed group at time point 3. So it can be argued that the mussels experienced an energy deficit at the third time point, necessitating an increase in sugars to feed into the glycolysis pathway, which can then help to fuel the glycolysis pathway where substrate level phosphorylation is possible, where ATP can be produced directly phosphorylated from ADP. Also, some of the metabolites at the top of the TCAs of the glycolysis pathway had similar metabolite levels in both the 16 degrees Celsius group and the 24 degrees Celsius group at the end of the experiment, so at time point 5, suggesting that an equilibrium had been reached between the depletion and the utilization of sugar-based metabolites in response to thermal stress. when we look at the TCA cycle, we see that in the control group at time point 1, the metabolites were higher than in the heat stress group. But then at time point 2, the heat stress groups showed metabolite levels similar to the controls or higher than the controls. So arguably, this suggests that there's an influx in ATP required, which is produced via the TCA cycle. Also, succinate had the highest concentration at time point 2 in the heat-stressed animals, and then we see a decrease at the other time points. So succinate can be used to support energy production via the reversal of the second half of the TCA cycle, where it produces NAD and FAD in response to heat stress, as seen on screen. A shift between aerobic and anaerobic metabolism is common for intertidal animals like mussels and in response to thermal stress, and it's also suggested in this study. So proof of this is seen via the production of lactate. So at the first 2 sampling points, lactate was reduced in the heat-stressed group of animals, potentially due to the depletion thereof to support ATP production. Yet an increase is seen in the next 2 time points, supporting increased demand to withstand the period of heat stress. So also the metabolite succinate is a common anaerobic metabolic marker in mussels, often associated with oxidative stress. Also, amino acids can serve as substrates or intermediates of anaerobic metabolism. So some of these amino acids peaked in the heat stress group. They peaked at time point 3 and 4, suggesting that resources have been utilized and requires an influx at that time point to sustain metabolic activity. Some metabolites can also be regarded as signals of oxidative stress, which is induced by the heat stress in this instant. These metabolites showed high concentrations at the first sampling point. So if we consider something like glutathione, this can act as an antioxidant, countering the heat-induced oxidative stress experienced in the heat stress group, so the blue group in this case, showing the highest at the very first time point. So taurine is also said to provide antioxidant functions in mussels, while the metabolite GABA has been reported to scavenge ROS during thermal stress. So when we look at purine metabolism, towards the end of the heat stress challenge, many of these metabolites were decreased to the lowest point across all sampling times. So at time point 5, this may have been utilized to predict these metabolites may have been utilized to protect the tissues against heat stress. Purine metabolism also supports energy supply, suggesting energy deficiency in the thermally stressed mussels. Also, pyrimidine metabolites were decreased at time point 4 and 5, suggesting utilization of these to produce nucleotides and subsequently increased protein synthesis. So to summarize the results, mussels subjected to a marine heatwave event utilizes sugar-based metabolites up to 48 hours post-exposure, whereafter an increase in glucose production is seen to ensure ATP production via substrate level phosphorylation, as well as in the TCA cycle. So in the TCA cycle, at 48 hours, we see upregulation of some of these metabolites to help to produce that ATP. Also, linkage to the anaerobic metabolism is a different method of which energy can be produced in heat-stressed mussels. It's also seen that these heat-stressed animals encounter oxidative stress at the first sampling time, with increased antioxidant molecules helping to scavenge ROS. And in the heat-stressed animals, we also see the depletion of purine and pyrimidine bases or metabolites. So ultimately, when a marine heat wave occurs, green lip mussels show capabilities to respond and acclimate to thermal stress by regulating the energy metabolism, balancing nucleotide production, and implementing oxidative stress mechanisms over time. This study hypothesizes that green lip mussels have the capability to respond to and survive short-term marine heatwave events, but the ability to rapidly acclimate to repeated events with longer time frames remains unclear and warrants future investigation. So what does this all mean for the aquaculture industry? So results like these can be used in selective breeding programs where physiological characteristics, which allows mussels to withstand these thermal events, can be passed down to the next generation of animals, creating in effect a more resilient and robust mussel. Also, data from experiments like these, like this and previous experiments can be used to mitigate measures such as moving marine farms into cooler waters, which can be seen as a responsible management strategy. And data like this can also be inputted into marine heatwave modeling, where it can help to predict mussel survival outcomes in the face of forthcoming marine heatwaves. I'd just like to thank everybody who was a part of the study, all the people on the ground, all of the collaborators and the funders. Thank you, Shane, for the opportunity to talk, and thank you all for listening to this talk. And lastly, if you wanted to know more about this specific study, it's been published earlier in the year within the journal Metabolites. And then speaking of metabolites, I'm serving as a guest editor in one of their special issues themed the application of metabolomics approaches and reporting standards in aquaculture. So if you have any nice metabolomics and aquaculture research, or you know of anybody else who wants who can submit something to this issue, please feel free to get in contact with me. Thank you. That's it, Shane.
Thanks so much, Leonie. It's a very awesome presentation, a very interesting results you showed there.
Oh, thanks.
Can you go back one slide?
Yeah.
So I just want to bring everyone's attention there to your email. Are you open to people contacting you to talk about more about what you're doing, collaborating about your talk as well? Are you okay with that?
Yes, definitely.
Okay, so there's Leonie's email up there. If you want to contact her, she says she is open to it. We're also going to put this onto our YouTube channel on Metabolomics South Africa. So anyone watching after this presentation in the future, I'm hoping that someone will be able to contact you. So there is the email. I think you can go forward one more slide just to show everyone the guest editor special edition.
Oh, yeah.
Yeah, so there we go at the bottom there. So anyone interested in joining in this topic, I know Sander is in the audience. He's a co-guest editor, and I think Andre was your previous co-supervisor from KZN.
That's right. Yeah, that's right. It feels like I'm talking to my family here.
So I'm going to open the floor to the audience. If there are any questions, you're more than welcome to raise your hand or have a chat in the chat box. Kit, do you have a question?
Yes, sorry. I don't know how to raise my hand, so let's just talk. I just want to know, Leonie, thanks once again. Good presentation. So just one question. I hate quick questions, but let me give you a quick question.
Yeah.
This is about, I know this is farmed animals and it is, well, animals deeper subsurface water. I just want to know, and I don't want you to guess about the environmental extremes, but in a natural shallow water on rocks, what do you expect will be the lethal temperature for these mussels? And I'm talking about the green lip mussels now, not Mediterranean and whatever type of, I don't know, mussel. But I thought I asked the question before Andre asked it. So did you do any other studies where you look at the lethal temperatures and time frames?
So in other studies, when the experimental temperature went above 26.5 degrees Celsius for like 4 days, at around day 4, we started to see mortalities. So that's about where the lethal limit is, so 26 degrees Celsius. So yeah, even in this one study we did, it was at 30 degrees Celsius, but only for 60 minutes. So that was just a stressor. But around 4 days, they started to die at 26 degrees Celsius.
Thanks a lot. Sorry, Andre.
Thanks, Beat.
I see Andre has a question in the chat. So he's intrigued by the GABA response. Is this also a neurotransmitter in mussels?
So we've done a few studies also where we placed the mussels in magnesium chloride as a relaxant, and that also had a big influence on GABA. So we also hypothesized that that's due to the neurotransmitting factor. So I would say yes, but it's not like I can 100% say, yeah, we've tested exactly with that in mind. But yeah, it does seem like it, yes.
So if mussels have GABA, which we have as humans, as neurotransmitters, does that mean they feel pain the same way we do?
No.
Ah.
No, I've never seen them shout when we experiment on them. So their stress response is very different, but they do synthesize that metabolite. That's true.
I know lobsters recently have been considered more of a sentient species. It's now illegal for any kitchen chefs to boil them alive as they used to, because they say they do feel it. So that's interesting, just maybe to think about at a crustacean level. Lobsters are not so far away from mussels.
But far enough, Shane.
Far enough. But if
Yeah.
you're having similar neurotransmitters going on there, it's something to consider.
True.
I don't know if, Sandra, do you have a question for your previous student?
What we're trying to do with our industry outreach, I mean, I've tried my hand here in South Africa contacting people like the aquaculture environment or just local agriculture, and try to actually, I want to say, share our science with them, our techniques, our way of looking at things, and those people don't bite. How is it different in New Zealand?
I guess it's a good question, Sander. They are a special part of society. They can be very stubborn as well, even in New Zealand. But the government plays a big role in it, and there's a lot of research institutes which job is literally to be a link between science and industry. So they've got a lot of working groups and meetings, which is it's almost compulsory for the industry to also be a part of that. So yeah, they get much more exposure to these things, and I guess scientists need to do the stuff too, and it gets reported back to government, which uses to make all the rules, which is then implemented to industry. So everybody is really interlinked in the process of doing it. So in the end, they are dependent on the science. So they're actually happy to listen. They're not necessarily happy to make the changes, but they're happy to listen.
Yeah, so that's like I said, as a scientist, we obviously have to make a difference in our community. So we're not close to the ocean, but I mean, at least at agriculture level, we can maybe, do some community engagement with the farmers and things around here. But yeah, unfortunately, it's still a struggle to get them to bite and to make use of our expertise. I won't say for free, but I mean, at least we want to help.
That is always the challenge, the politics. Are there any other questions from the audience? Francis?
Maybe a remark from me, Francis. Thank you for that wonderful presentation. I hope you can hear me. And
yes.
I was just wondering that there is the TCA anaerobic system, and I was just wondering the link between, whereas oxygen is not directly involved in that cycle, the availability of NAD, FAD is related to oxygen. And when temperatures go up, the amount of dissolved oxygen in water decreases. Would that have influenced what are the processes that we see that you picked in this analysis?
Absolutely. Oh, sorry. Yeah, definitely. So even though it is induced by heat stress, you're right in saying that it also affects the oxygen. And I mean, the mechanisms are in place in the animals to use stuff like that cycle in the absence of oxygen or with oxygen. So it's definitely linked.
Thank you.
I see Andre has another comment. It says, SA farmers are cleverer than academics. So what is next for Dr. Leonie?
A good question. I did my PhD on abalone, and we're now moving a little bit more to abalone research. So I think the mussels are parked at this point, but so next is more abalone things. But yeah, I'm always open for new opportunities and adventures. But for now, yeah, ticking along here. Yep.
Okay. Any other questions from the audience? If not, and those in the YouTube land that are listening post-meeting, we do encourage communication with the speakers. So do contact Leonie, either for collaboration of interest or even for the research topic as she is guest editing there. Thank you so much, Leonie, for presenting to us. I know it's rather late there at the moment. So thank you for joining us so late, and we appreciate it very much.
Thank you, Shane. Only a pleasure.
Okay, great. So have a wonderful festive season. I hope it is restful, and yeah, I hope things